to get started here, guys. Thanks so much for being here, everyone, again. It looks like we have a nice audience again here, which is really great and promising for our seminar series. Gosh. All right. Okay. Hello, everyone. So my name is Robert Driver. I'm a postdoc at Duke University. I want to welcome everyone here to another week of the AOS Student Research Grant Seminar Series. And before we get started, we'll introduce the AOS Student Research Grant, as we always do, and then the AOS Student Research Grant Seminar Series. So as you all know, I'm sure by now, the AOS offers funding to students and early career researchers with the AOS Student Research Grant. And each year, it's due right at the end of January, beginning of February. We receive around 200 applications, which is an amazing amount of effort to the committee that handles that, and also to the students and early career researchers who submit the grants. The grant has a finite budget, so it's a challenge for the winners to write a proposal that will advance the field of ornithology while still remaining feasible as part of that budget, which is often a graduate dissertation chapter, as it turns out. So the AOS Student Research Grant Seminar Series celebrates that challenging and innovative work of the AOS Student Research Grant recipients by sharing AOS-funded or related work to the AOS members. So as you guys know this as well, each week we showcase the research or research plans of a 2023 or 2022 AOS Student Research Grant winner in a seminar-style format. And so we've had one seminar each week for 15 weeks. We're in week 9 now. And I want to thank everyone for being here for our seminar, Lyn especially for presenting this week, and thank the AOS and AOS Student Affairs Committee for their support and input, and Ramona Fletcher and Chris Ann Handel for all their support each week. So before we turn our attention to Lyn's research, I want to mention to please put your microphones on mute. If you have any questions, there will be time for a Q&A session at the end of Lyn's talk, and you can participate by either raising your hand and I will unmute you and you can speak your question, or you can type your question in one of two ways. We have this Zoom Q&A feature chat box that you can use, or you can just type into the regular Zoom chat, and I'll be checking all these various ways of asking questions to make sure we get to everybody. And I'd like to remind everyone that this is an AOS sponsored event and that all participants are required to follow the AOS virtual meeting event code of professional conduct. And I will post a link to the AOS virtual meeting event code of professional conduct in the chat. And please be aware that we're recording this talk. In addition to the code of conduct, I'm going to post an email address that you can use to contact the Student Affairs Committee if you have any questions about the seminar, as well as a link to the post-evaluation form. And this is very important to get audience feedback, to know how we could improve the seminar series as we move forward week to week. Would really appreciate your guys' participation there. So I'll post those links just when I'm done talking. Okay, so today, Lyn Brown is currently a PhD candidate at Trent University, and Lyn's academic career began with a BS in animal behavior from Western University. These are all in Ontario, by the way. And then Lyn went on to receive her MS degree in sustainability at Brock University. And there, Lyn researched aquatic invasive species management in the Niagara area, and her MS research was published in Management of Biological Invasions. Prior to starting her PhD, Lyn worked at a variety of biological and internship positions, working with loons and piping plovers and Wilson's plovers and red knots, least terns, black skimmers, and of course her study species today, the American oystercatcher. And Lyn's final internship was at the Archbold Biological Station, working with that well-monitored scrub jay population, and her research from this internship was published in the Wilson Journal of Ornithology. In 2021, Lyn joined Trent University in Ontario in the lab of Dr. Erica Knoll. And continuing her research on American oystercatchers, Lyn has published on oystercatcher biology in water birds, as well as the Wilson Journal of Ornithology. In addition to the AOS Student Research Grant, which we all know, of course, Lyn won because we're at the seminar, Lyn received the competitive NSERC postgraduate doctoral fellowship and the Ontario graduate scholarship. In addition, in 2022, Lyn won the Jed Burt undergraduate mentoring grant from the Wilson Society, which is a great grant and shows Lyn's mentorship ability. And in addition, Lyn is extensively promoted and participated in outreach for the deaf. And I won't even be able to touch on all the work she's done for the deaf community. But in 2023, as part of the AOS Student Affairs Committee, Lyn organized a panel on the perspectives of birding and ornithology for the deaf, blind, and hard of hearing birders. And then Lyn created a deaf and deaf-blind accessible recording of the panel hosted at the 2023 AOS annual meeting in London, Ontario, I guess was that one. And with that, I will turn it over to Lyn for her talk entitled Diet Specialization in American Oystercatchers and Effects on Breeding Success.
Thank you so much, Robert.
No
And
problem.
thanks for joining everyone today. I'm really excited to share some of my research. Can you see the screen okay? Okay, awesome. So hi, my name's Lyn Brown, and I'll be sharing some of my PhD research focused on diets of American oystercatchers. And I want to start by thanking all of the people over the years who have helped conduct this research and provided support. It really has been a true group effort. A special thanks to my trusty assistant, Trevor, for all of his help over the years, as well as the staff at Chincoteague National Wildlife Refuge, who have been our main partners, as well as a number of partners along the Atlantic Flyway who are part of the American Oystercatcher Working Group. And the photos of the beautiful birds taken today were done by Trevor McLaurin, Ken King, and Michael Forgis. And I also want to thank all of the groups who have provided funding for this research, including the American Ornithological Society, for which we wouldn't be able to do this work without all of their support. So I'm truly grateful. All right. American oystercatchers are a beach nesting shorebird that breed along the Atlantic coast of the US primarily, from Maine to Florida and over to the Gulf Coast of Texas. They are considered a species of conservation concern because there are only 14,500 of them left in their population. And the state of Virginia has the highest concentration of breeding American oystercatchers throughout their range. However, since 2016, throughout the islands in Virginia, the population of breeding birds has been declining, and we're not sure why. These birds forage on a variety of prey, including mussels, oysters, clams, and razor clams that are found at bayside mudflats, as well as marine invertebrates like mole crabs and coquina that are found on oceanside shorelines. They provision their chicks from the time that the chicks hatch until they fledge at 35 days old, and continue to provision these chicks past fledging until the chicks reach 60 days old. And this is a very lengthy provisioning period compared to most other precocial shorebirds that are independent upon hatching. And it's because of the fact that it's difficult to open up a bivalve like an oyster. If you've ever tried to shuck an oyster with a knife, it's pretty difficult to correctly insert your knife, twist open the shell, and have the strength to do so, and then have the correct handling ability to then extract the flesh. And it's because of the time it takes for these birds to develop the strong enough muscles in their necks and their bills, as well as their feet, to correctly do this behavior, and then the handling to properly do it, that they have such a lengthy provisioning period. And it's because of this really long provisioning time that we thought there would be a relationship between food and breeding success. I do research at Chincoteague National Wildlife Refuge, which is found in the state of Virginia, and we work on two barrier islands, Assawoman and Assateague Island. And both of these islands are managed by the U.S. Fish and Wildlife Service to protect beach nesting birds, including the American oystercatcher. However, at both of these islands, since 2016, the number of chicks fledged per breeding pair was relatively low compared to the number of breeding pairs, which is the top graph, where we were seeing 20 to 40 pairs per island. And this low breeding success occurred despite management for their known threats, which included human disturbance and predators. So Assateague Island receives up to 1.5 million visitors per year that come to this beautiful undeveloped coastline. They allow pedestrian and off-sand vehicle driving access on the oceanside shoreline. To manage for the high level of human disturbance, which can cause birds to abandon nests or fully avoid using an area, the refuge closes the nesting portion of the beach, which you can think of as the higher berm area where a bird would nest. Even though the nesting areas are closed to benefit the birds, vehicle and pedestrian access is still allowable in the ocean intertidal area where birds might forage. The refuge also traps for the known predators of foxes, raccoons, and opossum, and these birds also experience avian predators like gulls, crows, peregrine falcons, and bald eagles, as well as ghost crabs, which will tunnel underneath a nest and can take eggs or small chicks. So despite the management for the predators and the human disturbance, the breeding success remained low, and we weren't sure why. So we investigated if foraging factors affected breeding success. To tackle this question, we first had to determine what the birds were eating on the islands. And I'll go over how we study foraging using a combination of behavioral observations and GPS tracking. For our behavioral observations, we do 10-minute observations on foraging adults or hour-long observations on provisioning chicks, where we pick a focal animal and we record exactly what prey species they eat, how much, and how much they feed their chicks. So we're visiting the mudflats or oceanside shorelines where these birds feed. And when we're recording their foraging behaviors, we also record their unique color leg bands so we can link their foraging to their breeding behaviors. And you can see one of those leg bands in the upper right photograph with the green colored band there. We also capture our birds to put on GPS trackers so we can study their fine scale foraging movements. And to capture our birds, we use leg loop carpets. Those look like chicken wire in the left-hand photo with monofilament loops, so clear fishing line tied around them. And these are placed around the nest site and buried under the sand. Then we'll put a pair of decoys and play the bird's alarm call, which if you haven't heard, sounds something like this. And these birds are highly territorial, so if they think an intruder is at their nest site, they will come to try to remove the intruders and hopefully get caught in our traps. So I have a video showing what this looks like. Originally, the nest was within the tire. We removed the eggs, put them in a padded box in the shade, and then we buried our traps around the tire, placed the decoys, and played the alarm call. And you see that our pair, both the male and the female, come in doing their piping display. This is where they're bowing their heads and approaching the intruder. And this is about as aggressive as it gets for an oyster. During this territorial display, sometimes the birds will trip on our traps, and that's when we're able to. You'll see the bird on the left will trip right there, And so that's when we might have been able to capture them. When we get our hands on the birds, we would safely disentangle them from the nets and then put on their GPS trackers. So these are Path Track Nano Geofix RF tags, and they take a GPS fix every 15 minutes at a 10-meter spatial resolution. So they will pinpoint exactly which oyster reef or mudflat a bird is feeding at. And these tags are attached with leg loop harnesses. So if you think of a rock climbing harness where you would step into the harness, that's how the bird is getting into its harness. And then there's a little bit of foam padding so that it's not abrading the feathers. And these tags do not negatively impact their movement whatsoever. And I'd like to thank AOS again, because this is where some of the grant money was going to buy these tags, for which we were able to understand their foraging movements. And while we have the bird in hand, we also add their colored leg bands so we can distinguish individuals, and we take some standard metrics of the bird, including body mass and their bill length, which I'll talk about later on. To download the data from these tags, we use a remote base station, which is shown on the left. So when this base station is within sight of the bird, usually within 200 meters, it can offload the data using Bluetooth frequency. And the data that we get from these birds is shown in these animations. So we had deployed 10 GPS tags in 2020 and then we redeployed them again in 2024. So on the right-hand map, we have Assawoman Island, and each of the colors is a different tag birds. And all of the birds on Assawoman Island show the typical oystercatcher foraging strategy, which I call a generalist. So they are traveling up to 2 kilometers away from their beach nesting site to forage at both the bayside mudflats and marshes, as well as the oceanside shoreline. In contrast, if you focus on the left-hand map, especially the 2 central individuals in green and pink at the bottom, these birds show a very different foraging strategy where the birds stay very close to their nests, often within 300 meters, and eat almost entirely one prey species known as the mole crab. So this is the first case of diet specialization that's been described for this species. And our behavioral observations match the GPS tracking data. So from 2022 to 2023, we made 816 foraging observations and chick provisioning observations. And we found that the mole crab specialists are eating mole crabs more than 90% of the time. And that's shown as the dark green bar in both graphs. They also eat a little bit of coquina, which is another marine invertebrate found on the oceanside shoreline. In contrast, the generalists eat mole crabs plus 6 other bivalve species that are mostly found on bayside mudflats. So this brings up the question of, why are we seeing diet specialists on Assateague Island? And to understand this, I went ahead and mapped the nests of birds based on their diets. So red dots are indicating specialists and black points indicate generalists. So you can see that all of our specialists are occurring on the left-hand map representing Assateague Island, and they're mostly found on the southern end of the island. In contrast, the right-hand map of Assawoman Island only has generalists. And we think that diet specialization has to do with the distance that a bird travels to Atlantic ribbed mussels. And Atlantic ribbed mussels are an important food source for American oystercatchers because optimal foraging theory predicts that this should be the most optimal or most profitable food that a bird should eat because it has the highest food profitability, which is the amount of food, its food weight, divided by the handling time, the time to capture and swallow the prey. So when birds have close access to Atlantic ribbed mussels, as on Asswoman, where all birds can reach ribbed mussels within 280 meters of their nest site, they will eat a combination of ribbed mussels plus other prey that are found on the bayside, as well as mole crabs and other invertebrates found on the ocean side. While on Assateague Island on the left-hand map, these birds can still access ribbed mussels, with the farthest rib mussels being within 3 kilometers of their nest site. And this is still within a reasonable foraging range for oyster catchers, which we've tracked traveling 16.5 kilometers away from their nest site to forage. However, when the rib mussels are further away, the birds instead are opting not to forage on the rib mussels and are instead turning to the second most profitable prey species, the mole crab, and forage almost exclusively on it. So, diet specialization has some adaptive significance for adults, where the birds are staying very close to their nests to forage, and this can conserve their energy. And this is important for a long lived species like the oyster catcher, who on average live 10 to 12 years, but can live up to 20 years or even longer. So, by conserving their energy to forage, they can have more energy for future breeding seasons. Additionally, by staying close to their nests to forage, the birds can simultaneously forage while watching over their chicks to protect them against predators. So, this potential for improved predator vigilance might improve chick survival. We also wanted to check if diet affected the ability of birds to meet their chicks' energy needs based on our behavioral chick provisioning observations. We could calculate the caloric energy intake that was delivered to chicks, and there was no difference in the caloric energy delivered to chicks based on diet. So both strategies are equally effective at meeting chicks' energy needs. Similarly, both diets are equally effective at meeting adults' energy needs, because generalists and specialists had similar body condition, which was their body mass given their wing chord length. And both were efficient foragers because they had similar food profitability. But taken together, the knowledge of some birds who specialize feed on mole crabs can be important for conserving the foraging habitat for these specialists, where the refuge might want to consider not only closing the nesting areas of the beach to benefit the birds, but also the oceanside foraging areas where the mole crabs live. And mole crabs, similarly to oyster catchers, avoid human disturbance. So, to create healthy mole crab populations, that's why they would want to close that oceanside shoreline areas. And also considering management activities like beach renourishment, where they're piling new sediment on to build up the elevation of the beaches. This might disturb the sediment and depress the number of mole crabs. So trying to do these activities outside of the breeding season would benefit the birds and the mole crabs more. And this type of management for healthy mole crab populations can benefit other birds who also feed on mole crabs, which include the U.S. federally threatened red knot, as well as ruddy turnstones, sanderlings, and willets. So I'm going to bring this back to the original question of how foraging factors might impact breeding success. I talked about how we measure foraging. Now I'll talk about how we measure breeding. In the state of Virginia, the breeding season is typically from April to August, with the birds starting to lay their eggs next month. Typically, clutches will have up to 3 eggs that take 28 days to hatch and 35 days to fledge. So myself and my technician Trevor, as well as the team at Chincoteague National Wildlife Refuge, would visit nests 2 to 3 times per week, and we would record the number of eggs and or the number of chicks, as well as record signs of nest fate, including if they'd been depredated or if the chicks were dying of starvation or a flood had come and washed away the nest. So we combined the information on their breeding with the foraging information I described before. And specifically, we looked at 3 factors that might affect the daily survival of chicks. And those foraging factors included diet, the distance to the foraging area, and the number of provisioning trips that chicks made. And we wanted to include distance to the foraging area from the nest site because previous studies on American oystercatchers as well as Eurasian oystercatchers have indicated that when the foraging area is closer to the nesting area, birds have higher breeding success. And it's because they might be able to make more provisioning trips to the chicks, and they spend less time away from their nests, so they might have heightened vigilance against predators. So we took all of that information and ran a chick survival model using the program MARC. And overall, we found that none of the foraging factors affected daily chick survival. So, what I interpret this as is luckily at our site, food is not a limiting factor for breeding success for these birds. We were not finding chicks dying of starvation, and all of the chicks were able to gain a healthy weight, and the adults were able to deliver enough energy to them. Instead, the limiting factor was that older chicks tended to have lower survival rates compared to younger chicks. And this might seem counterintuitive at first, but we found that the older chicks started to become bolder and hide less from predators. Additionally, the older chicks were of larger mass compared to younger chicks, which could represent a larger food reward. So we found that 3 to 5 week old chicks were getting taken more by avian predators like greater black-backed gulls compared to younger chicks. So this diet specialization research is really interesting because there's a lot of spin-off research that we've been able to do from this. And this is because the Eurasian oystercatcher has a really rich history of diet specialization research. So in Europe, there are some Eurasian oystercatchers that specialize feed on worms, and those birds tend to have longer bills that are able to more more easily access the deeply buried worms. There are also Eurasian oystercatchers that feed on cockles. And cockles are similar to mussels where they have a hard shell. And those birds tend to have shorter, more robust bills that can more easily crack open the hard shells. So there's a relationship between the bill size directing what bird or what food a bird might more easily be able to eat, as well as the food can actually reshape the bill over the course of 6 weeks. So we wanted to see if the specialists and generalists differed in their bill shape. And this is preliminary data still based on a sample size of 12 birds from the past field season in 2024. But so far, we're not seeing a difference in bill size between specialists and generalists. And this is a good reminder that generalists also eat mole crabs. So their bills can't be too different to continue to eat the mole crabs. Additionally, we're not sure what the specialists are eating in the non-breeding season, so they might be switching their food source, which would reshape their bill. And so this is some other research we want to continue to look into in the future, where with the winter tracking data that's still coming in and some prey data that we took over the winter time, we might be able to start to figure out if the specialists remain specialists year round. And some other research that has come out of the diet specialization is this unique feeding strategy on mole crabs that both American oystercatchers in Virginia as well as whimbrels out on the Pacific coast of Oregon have been observed to do. And this is work along with my collaborator, Seabird McKeon, who is based with the American Bird Conservancy, and this publication is available with the Wilson Journal of Ornithology, and I have a QR code there. So I'll show you a video of what I'm talking about when I discuss egg stripping of mole crabs. And I'd like to point out the eggs of the mole crabs in the photo pictured here. So those are those orange bodies under the talsum, the tail of the mole crab. So in this video, we had an oyster catcher that just grabbed a mole crab. Now it flips the mole crab on its back on the carapace, and it will selectively remove the egg from the mole crab and discard that carapace. So based on our 2022 data, we observed 554 mole crabs that were eaten, and this egg stripping behavior occurred 10% of the time by 11 individual oyster catchers. And Seabird observed this 41 times with Whimbrel out in Oregon. We both found that birds were targeting larger mole crabs to strip their eggs. And this makes sense because only at a minimum size of 10 millimeters do female mole crabs start to produce eggs, and larger mole crabs tend to carry more eggs. I did not see an association between mole crab diet specialization and this egg stripping behavior. So what's going on with egg stripping? We think this has to do with digestive bottlenecks or the digestion rate model. So if you think of the bird's stomach as this funnel, as the birds begin to feed on mole crabs, they will swallow them whole. But as their stomach fills up, they want to continue to be able to eat and gain a lot of nutrients without taking as much space in their stomach, so they don't have to pause to digest. So that's when we think that they're switching to just extracting the eggs. And this has some other research that we want to look at in the future, which comes after the mole crab's eggs are stripped by the oyster catcher. So again, I'm just going to let this video play. So the bird got a mole crab from the surf, it flipped it over, and will now extract the eggs. And I want you to watch what happens after this behavior occurs. The carapace is discarded, and you can see there are a number of other small peeps circling the oyster catchers. We have some sanderlings and some ruddy turnstones. And now we have the successful ruddy turnstone who has grabbed that mole crab carapace and will feed on it itself. So we think that the oyster catchers are supporting feeding commensalism with ruddy turnstones and sanderlings who come along and eat their discarded carapaces after the eggs are stripped from the mole crab. And this is ongoing research where we will be doing more concentrated focal sampling of ruddy turnstones and sanderlings to see if they have higher foraging rates on mole crabs when they're following an oyster catcher that's stripping the eggs compared to when an oyster catcher is absent. Some other research that I did during my PhD was looking at the diet overlap between male and female American oystercatchers, and this has been published in Waterbirds, and I have a QR code for that publication as well. And this behavior is interesting when taken into context compared to other oystercatcher species around the world. So most oystercatcher species showed totally different diets between males and females. This occurs for pied oystercatchers and sooty oystercatchers in Australia, as well as African black oystercatchers and Eurasian oystercatchers. And the sooty oystercatcher shows the most extreme example in diet difference between males and females, and the most extreme difference in their bill lengths, where females have a 19% longer bill on average compared to males. So we wanted to see if male and female American oystercatchers might show different diets, since this pattern has held for other oystercatcher species. And male and female American oystercatchers also show this difference in bill length, where females tend to have longer bills than males. So based on our 2022 data of 28 females and 31 males, we saw almost identical diets between the two with a 99% overlap. So what's going on with the American oystercatcher species compared to the other oystercatchers? We think that it has to do with differences in the competitive environment, where American oystercatchers, if you remember, the study population I have has a very low breeding population. So there aren't many individuals, and so this would relax the selective pressure to reduce competition by having different diets. In contrast, the other oystercatcher species who showed very different diets have much higher competition. So the Eurasian oystercatcher numbers over 1 million individuals, and not only do they have to compete for food with other birds, but also with the shellfish industry, which has caused major collapse in the number of cockles in recent years. Similarly, the pied oystercatcher and sooty oystercatcher also occur in highly competitive environments. The African black oystercatcher is an interesting case because originally it was reported that males and females had very different diets, but in the early 2000s, a new Mediterranean mussel species invaded the area as an invasive species and became an abundant food source. And then they started to report similar diets between males and females. And similarly to our study, black oyster catchers at the Pacific coast of North America report similar diets between males and females, and they also occur in low breeding densities with sometimes less than one pair per square kilometer. Now I'll switch gears to some other research that we will be starting next month and for the next 3 years. So we'll be expanding the diet research of American oystercatchers to other bird species at the study site in relation to human disturbance. And this is because the refuge will be relocating a 1,000 car parking lot that supports that 1.5 million visitors to an area with low shorebird activity. So this gives us an opportunity to look at the effect of that parking lot relocation and the removal of human disturbance on the breeding and migratory shorebirds. And this is work that will bring on a new collaborator at Trent University, Autumn Watkinson, who specializes in threatened avian species restoration. And it's already been a huge collaboration with a number of groups along the Atlantic Flyway, for which I'm immensely grateful for all of their support and resources and funding that will make this research possible. It really is a huge collaboration. So to provide more context about the human disturbance and some of the other shorebird species we'll be studying, the refuge is considered a protected site within the Western Hemisphere Shorebird Reserve Network because of its importance for breeding and migratory American oystercatchers, breeding and migratory piping plovers, and migratory redknots and whimbrel. And this high bird use also overlaps with the high human use. So I mentioned that the refuge on Assateague Island receives up to 1.5 million visitors, and they're primarily coming to a 1 kilometer stretch of beach, which is supported by a 1,000 car parking lot. Immediately adjacent to this parking lot is a 5 kilometer stretch of beach, which allows off sand vehicle driving. And this high level of human disturbance doesn't mix well with the birds. I mentioned that the refuge closes the nesting portion of the beach for the birds, but even with a constant stream of people, these birds have very low tolerance to people and can abandon their nests. More often, they're just totally avoiding using otherwise suitable habitat for nesting. And similarly, once the bird's chicks hatch, they are mobile and they can leave the protected closure and enter that oceanside foraging area where they're coming into contact with people. And this is often leading them to avoid using the high quality foraging habitat, wasting energy running away from people, or totally avoiding using an area. And we see a similar case with the migratory shorebirds, like the red knots, who are traveling thousands of kilometers from their wintering grounds at the tip of South America to their Arctic breeding grounds. So when they stop over at these sites, they are at critical reserve fuel levels and need to spend all of their time resting and foraging. However, they're also low tolerance to human disturbance and will end up wasting their energy, flying away from the people, or totally avoid using the high quality areas. So luckily, after 15 years of planning and $18 million, the refuge will be moving the parking lot, which is shown in red, to a site that's 2.5 kilometers north and has very low shorebird activity. Once that move happens, all access to the public, including pedestrians and vehicles, will be shut down during the breeding season from mid-March to mid-September. So this presents a huge opportunity to look at the effect of that parking lot move and the removal of human disturbance on the breeding and migratory shorebirds. I will just briefly touch on what we're measuring for this, but I'm happy to talk about this more with questions. So this is expected to be a 3-year study. We'll start collecting data this month in 2025 to get the pre-relocation data. We have years of pre-relocation data, but this will get us some more specific data. And then we'll do 2 years of post-monitoring after the construction is done by 2026. So for the migratory shorebirds, we will be counting the number of birds and their foraging habitat use, as well as their prey communities in relation to the parking lot relocation. And for the breeding birds, we're looking at their overall numbers, as well as their nesting habitat and nesting predators and nesting and breeding success. And for the oyster catchers, we still have those GPS trackers. So we'll look at their home ranges and habitat use, where they might start expanding their home ranges to areas that previously had high human use, or switching their foraging activity to be more during the daytime since they don't need to avoid the people. And the relocation itself is expected to restore 6 kilometers of prime nesting and foraging habitat for shorebirds. And the research should be quantifying the conservation investment of relocating that parking lot, as well as the benefits to the birds from the removal of human disturbance. And specifically, the refuge plans on using this information to continue to justify why they need to close off beach access during the breeding season after the parking lot moves. In case you haven't guessed, it's taken 15 years of planning because there is still some local resistance. So they expect that after the parking lot moves, the public will ask why they can't have 2 parking lots and more beach access. And so we're expecting all of these benefits once the humans are removed from the area, and so this research could provide rationale for why they need to continue to close off beach access. Similarly, the refuge wants to understand the sort of management and restoration activities they might perform after the relocation. So for the nesting habitat, it's a clamshell substrate parking lot, which tends to be the preferred nesting conditions for plovers and oyster catchers, and the refuge wants to know if they should continue to dump sediment in that area to benefit the birds this time, rather than the goal being to create a parking lot. Similarly, should they start to manage their dunes for the birds rather than build up the dunes to protect the parking lot infrastructure? Do they need to do certain vegetation management to create more preferred nesting habitat for these birds? And they're always working within confined funding. So currently, they trap mammalian predators to benefit the nesting birds, but once the people move northwards, the trash and predator attractants will likely move northwards. So they want to know if they should redistribute their predator control efforts or even can scale back on them. And some other research I wanted to point out, which Robert had introduced, was the deaf birders panel that I helped produce at the American Ornithological Society conference. I have a QR code with the original recording. This is a wonderful resource that was put together by a great group of people. We were interviewing undergraduates to establish career professionals on their take of what it's like to bird, as well as barriers to access and how to dismantle those barriers. And we're currently working on producing or publishing a set of guidelines to help inform the hearing community. So please do check out this resource. It's available on YouTube on the AOS's website. And that is all I have for you today. I have some additional links if you want to learn more. The top one is our research website. If you're curious about more of the field work, there's a podcast interview, and I have my contact information below. So thank you again for listening, and I'm happy to take any questions.
All right, Lynn, thank you so much for the great talk. Uh, Lots of wonderful visuals. I think I speak for the whole audience. We enjoyed that oyster catcher getting trapped. I'm sure everybody enjoyed that. And thanks so much for your work with the deaf as well. Chris Ann has actually posted the YouTube link in the chat as well. So if anyone wants to click that link, they can access it that way. And people are saying excellent talk and things like that in the chat. And if anyone has questions, you can place them in the chat or the Q&A. If you're saying good job and stuff, just try to click everyone. I guess Lynn can also see the hosts and panelists if you click that as well. But thanks so much to everybody for being here, and I'll look out for questions. And I'll just start it off. I wanted to ask, is there anywhere about the bill size differences? Is there
Mhm.
anywhere in the American oystercatcher range? I saw it's on Hispaniola and maybe the Bahamas, if I'm remembering this map correctly. But regardless, is there anywhere in the range where it's more densely populated that could have a bill size difference?
Yeah, that's a great question. So I am most familiar with the oyster catchers within the US, but even within the US, the northern regions are doing better breeding wise compared to the southern areas. And we're still not really sure why. The north, where and by north I mean like the New England states, New York, that sort of area, their breeding productivity, so the number of chicks fledged per breeding pair, has been well above the target of 0.5 chicks in the past decade for sure. And they have a lot higher human disturbance levels there. And like for example, within Virginia, almost all of the barrier islands are protected either by the US Fish and Wildlife Service or the Nature Conservancy or the state. And so they're not experiencing that threat of human disturbance, except this one island where I've mentioned we have the over 1 million visitors. And they're across the US. in the protected areas, they're generally managed for predators and human disturbance, but they really don't know why there's been this steep decline in the more southern areas in the past decade. But in comparison, yes, areas in South and Central America are seeing much higher numbers. So it would be interesting in the future to look at if there might be differences in bill length and diet there.
Great. Yeah, I'd be interested if it followed the other species in some parts of the range, but not others. And then Julie Marr asks, when doing the 1 hour behavior surveys for chicks, was there a certain time of day that this was conducted?
It was pretty randomized throughout the day and throughout the tidal cycle as well. I think for these birds, the tide is actually the more important factor rather than time of day, because the falling tide and low tide is usually when the birds have better access to those bayside mud flats. And then just after high tide, during falling tide, is when the mole crabs come to the surface more and are more easily accessible. And this, we were out there at all times of day and throughout the tidal cycle.
And Kate Goodenough asks, I think, a related question. How much of the foraging activities occurred at night?
Yeah, great question, Kate. And I'm just starting to dig into that data. As you might remember, Kate had helped train me on the GPS trackers and was so gracious with coming out to our site and helping us get started. So, uh, the first round of GPS trackers, we actually turned them off at night to save battery. But with the upcoming beach relocation project, one of our goals is to look at if the foraging activity will switch more from the nighttime to the daytime. So we will be doing more concentrated effort looking at that question. So I don't have an answer for you now, but I will in the future.
And I'm going to ask another question here. In the beginning, uh, you described the difficulty of like opening an oyster, shucking an oyster, and how the
Mhm.
birds have to work to do that. So in these birds that are to teach the young and how that increases the provisioning period. So in these birds that are not accessing oysters as much, that are more reliant on the ghost on the crabs. Is there a change in the provisioning period then if it's not as complicated?
Like the length of time that the chicks are provisioned for, is that what you're asking?
Yeah, I guess I'm wondering if the oyster is a complicated food to uh, eat and open for these birds that are not opening or eating them regularly. Does that change the provisioning period? Yeah.
Yeah, that's an excellent question. And I think in the future, I'd probably have enough data to look at that further. We don't tend to do many observations after the chicks fledge at 35 days old. And uh, you have a much fewer or a much lower sample size when the chicks are getting to that age because so many of them are picked off by predators. But we still they're sort of like the teenager or older child that you can't kick out of your basement and is just hanging on and taking energy and effort away from their parents where there we still see chicks that are 40, 50, 60 days old begging from their parents even though they can um eat themselves. So yeah, that is a really good question that I'll look into more in the future.
Cool. Yeah, making me think about humans a little bit with that answer. Uh, and so we have a question from someone anonymous. Have you followed the diet of the young chicks, and are they generalists or specialists when they leave their parents in your area of study?
Yeah, that's a great question as well. So that's something I think we could start to answer more in the future. When I originally started at this study site, only 20% of our population was banded, and those were the adults. And we don't actually band our chicks just to minimize the amount of disturbance. But there are other places along the flyway where they do ban their chicks, and now there are other collaborators who are collecting GPS tracking data, so might be able to look at this question of specialization and if that carries over to the chick, or is heritable or whatnot. And so these are excellent questions for the future. We just don't have the data at the moment.
All right, and I'm just checking the various methods to ask questions, chat, QA. If I miss one, just let me know. But I think I got everybody, and we are pretty much right at roughly 1 o'clock. So uh, just again, thanks everyone for being here, and thank you so much, Lynn, for the excellent talk. I really learned a lot about oyster catchers that I didn't know before. And again, of course, the great videos. And thank you, Chris Ann. I actually had it copied onto my clipboard, but I didn't paste it. This is another opportunity for people to fill out the post-seminar evaluation form if you're an attendee, to give us feedback. And I think with that, we'll end the seminar, and just thank you so much, Lynn, again, for sharing your research with us. So.
My pleasure. Thanks for the invitation.
All right, guys. We'll see you next week.